Sorry. All right. Just make sure that it's recorded. So if you didn't hear me clearly, please let us know through the chat box as well. So, our technical can assist and helping you with that. All right. But I think right now it works well. All right. So once again, my name is Suppakarn JANDANG. You can call me May. I'm working at the Center for Ocean Plastic Studies. And my part today is about the monitoring techniques. So before you start to do anything, your research or even your own daily life, so you will start with the questions. Actually, it's very simple. For the monitoring designs, you can start with the WH questions. Very simple. What, when, where, and how. And then if you get the answer, you will get the good designs of your research. But in my opinion, this kind of question is kind of, like inside of the process. But what is the real questions at the beginning before you do in your research? I think this is very important. The why questions, because the why questions can clarify the purpose. To help you to clarify your research, it forces the researcher to articulate the reason behind choosing the particular topic or questions, and also define your problems. Help to define the research problems, research aim. You can address the research aim as well. And also, eventually, that will guide you to the research design. And as you can see from this slide, the why questions can come up with your goal. When you have the goal, it's very easy to follow and plan later. So for example, the why question and comes up with the goal. You can, I shared it. Oh, sorry. Is that this one? Sorry, guys.
That's
Did you see
okay,
it?
Yeah, now we can see it. Thank you.
Sorry, I will not repeat it again, but yeah, this is just what exactly, what is the question to begin your research. So yeah, where am I? So yeah, the why question can setting you a goal, so then you can follow your way and you can plan your methodology later. All right. And what is about the other questions that you have to ask before you're doing your research? I'll start from the what you're gonna do with the microplastic. Before I start that, I checked roughly for the registered audience for this webinar. It's kind of half that some of them they have experience and some of them didn't have experience about microplastic. But today I didn't give you any definitions of microplastic. I think you already know what is a very tiny particle that is contaminated around our environment. But yeah, later you can get into the deep details about this. But if you have any questions, please do not hesitate to let us know. The first question, what you gonna do with the microplastic, what you gonna investigate. For example, in our current work, we would like to investigate the abundance of microplastic. Some of them even try to look at the type and origin, even the what kind of polymer type that is can be dangerous to organism, or even the distribution of microplastic. And eventually, it can be impact to the biota, which I will say later. And when you will do your research, you have to determine when is the best. For example, if your plant, you would like to see the difference between wet and dry season, you can choose, like to investigate both of their season, the tidal, the period. All of these factors can affect your result and your research. So make a good plan for this. The last one, I put a specific phenomena. As I I'm just currently working with the coral reef as well. So right now, as you may know, is kind of, the huge problem with the mass bleaching event. This kind of we call the specific phenomena. And then this is kind of thing that you need to investigate more in terms of, environment and the biota. Where? In terms of the where, it's not meaning that you will choose like Thailand, Singapore or whatever. In terms of where you going to collect, actually there is some metrics that you can do your research. As you can see here, the water. If you're talking about water, the seawater is not just only the surface water, there is a water column. So which, depth that you would like to do your research, shoreline, seafloor, or even the biota. I saw some participants that not working just only, the marine environment. Also, they're working on the terrestrial as well. So I would like to clarify one thing first. So this webinar actually we focusing on the ocean plastic studies. So the methodology, the equipment, all the metrics. So we will, like talking mostly in the marine environment. So sorry about that, but I really hope that you can apply our, uh, the equipment or something that I'm going to share you later for your own research. All right. And how? You better define the spatial or temporal scale. What is you going to do with your research? And please use the repeatable protocol. Why is that? Because the repeatable protocol is help you to ensure this protocol is works well, and then you can compare your research to others. Actually right now we are facing a problem, which is that uh, many of us around the world, they are doing their own research, but they didn't consider about the data comparison or the data harmonization. So this is a gap that we have to fill it. So better plan your research and then using the unit or the protocol that you can further research can be collect your data and then for the data harmonization to get a big data, we can see the trends or something that we can do some further analysis as well. Overall, to plan of these things about the plastic pollution research, we would like to achieve the SDG goal. As you know, we are currently face many kind of pollutions. So we encourage you all to, doing the research and tackling the plastic pollution and to achieve this goal. All right. Before I going a little bit deep down to my parts, I would like to remind you one thing before you doing your research, please clarify the unit that you will use. For example, the sediment, what kind of currently unit that people use. Actually, this is a problem from my own experience as well. For example, I'm working with the corals, there is some challenges because there is no standardized method or even the harmonizing the data. So right now it's just like a, like a dot. We cannot connect yet. So if, for example, if you're working with the floating microplastic, they already have a data harmonization, the standardized, so you can choose from that. Actually, if you really know the unit of your samples, that is could help you to, can get the result easier, even compared to others' work as well. The metadata. So before you doing anything, please record your survey ID. Some of you may have, once you go enjoy with the cruise or something, you have a survey ID. The location is very important. The date, the equipment that you use during your sample collections, and even the environmental variables, because in each countries or each season have this kind of variable could affect your results, and then you can clarify with this later. All right. Okay, so in my part today, I'm going to share with you about the sampling techniques and the method, in terms of monitoring. And we are focusing on three metrics here. First, the water, second, sediment, and the third is biota. For the floating microplastic, all around the world, they conducted this kind of research using a net. As you can see, we have different kind of nets, Newton net, Mentor net, even the plankton net. You can collect the sample using those kind of net. It depends on what available in your lab. So, but basically our center is using the Newton net because it's kind of, easier assembly and design, and it can put, like flow meter and everything. Maybe we are familiar to this, but maybe if your laboratory have a maintenance, please go ahead to do that. And why? I will show the reason why the net is very good for collecting the floating microplastic on the surface. This is the reason of the advantage of the net sampling. If you need to understand more in terms of detail, you can go for this guideline, the guideline for harmonizing the ocean surface microplastic monitoring method, which is wrote by Ishida et al., in the previous work, and you can still follow this method. Why the net is very advantage for conducting the floating microplastic? First, they can collect the large volume of the water. If you use another kind of, for example, the water sampler or something, you can collect with that as well. But, comparing to this kind of net, you can have the large amount of the water. So, and the net also easier to deploy compared to a pump or a little bit more complicated machine like a CPR, like a connected plankton recording, something like that. And the previous work, how is benefit to compared to the previous work? Because many, as I mentioned before, many of researchers, they conducted collecting the microplastic surface using a net already, and then you can compare with them. And this is kind of the harmonizing data as well. And yeah, this is just a sample how you assemble your net to the boat. As you can see, I mean, some of you may didn't have any board or something, but we can apply. If you have any questions about this, let me know later. Okay. First, you have to understand the behavior of the microplastic. If you cannot imagine, I suggest you to try to think about the Pacific gyre. Some of the microplastic behave like that. Their behavior is just like that as well because they are floating as a patch. So the large volume are needed because, for example, if you collect with the bucket or something, if you just collect in the patch of the microplastic, it could, affect your data. So using the net could help you to, get rid of this problem. And then the duration times of the towing the net, we suggest to use like 10 or 20 minutes to avoid the clogging. When you use, like a small mesh side of the net, so it's very easy to clog, especially if you are interested to conduct your research in the coastal area. The speed of the boat, we recommend to use just only 1 or 2 knots, just only to avoid extrusion. If you go very fast, so everything, gets mixed up, and then it's very difficult to deal with, actually. And the net immersion depth, it will be like half or third-fourth of the height of the, square, the net. Buoy and weight need to be stabilized the nest as well. As you can see from this photo, the yellow one is a buoy. So this buoy can help your net to be stabilized, and then you can continuously collecting the microplastic from the surface. All right. The water volume, So as I mentioned, the net can collect a large volume of the water. So we normally receive this kind of questions because some of the laboratory didn't have any flow meter to attach with the net because they didn't working any with the plankton before. It's happened in the real situation. So they asked us like, is it necessary to not to use is that necessary to use a flow meter? Actually, it's not. You cannot use a net without the flow meter, but actually, if you have, it's recommended because it's quite more accurate. You don't need to convert, the unit or whatever later. So, but if you really didn't have it, so it's okay. You can still conduct your research. For who that have the flow meter, first, when you attach to the net, you have to do the calibration. Yeah, this of course you have to do it, otherwise, it will be not correct data. And then please take a note of the number before and after the net drawing, because you can use this number to calculate later. And some of you may think like, how like the distance of the net go. So the estimate actually is not exact, but you can use the GPS, to estimate the volume and the area, the distant area of the net showing. So this is kind of some simple equation that showing you and how the net will be immersed underwater. So basically, the net will be like half or third fourth underwater, not like submerged into the water. Otherwise, you cannot take anything about the floating microplastic. All right. And what you're gonna do once you already collect the sample through using the net. This is kind of onboard sampling processing. I mean, if you really have a vessel, it's very helpful because they have that kind of supporting equipment. You can work is easier. I will share with you about our experience, working on board with the sample processing. As you can see, this is a Newton net. When we collect all this microplastics, so we fix them, and then using the hose to wash um, microplastic or if any particle that attach inside of the net from outside, using a hose that already prepared on the boat. And just remember this to avoid any contaminations, because we use like the seawater to wash it and not pass through any filtration. So make sure that you wash it from outside, not inside of the net. So otherwise your sample will be contaminated from other seawaters. And another tip is always avoid using a plastic tools. I know it's very difficult, especially when you working on board, because you think plastic could be easier to carry or to keep it. Yeah, totally agree with that. But I think in terms of the water samples, so you can choose another kind of a polymer type of plastic, for example, the high density, such as PVC or PET. The reason why is that because actually the floating microplastic, like in the sense it's supposed to be the lighter density, so it's quite difficult to contaminate with the high density. But if you conduct your research in the coastal area, is that something else? You have to consider which is the best condition for you. The glass container or the bottle is also you can use, it's really safe, but it's fragile. It can have an accident with on the boat and quite dangerous, or even other materials, stainless, metals or whatever. All right. Once you, let's get back to our sample. Once you collect, all of the particle into the cord end of the net, and then you will rinse again through the handy net as you can see, it is easy to follow for the picture from this figure. So you rinse all of the particle through the handy plankton net, and then wash and rinse again. Collect all the sample in the bottle. After that, what you're gonna preserve your sample. We recommend to use the formalin, 4% formalin, to preserve your samples. Otherwise, they have many kind of reaction is could occur and then could affect your microplastic samples. So fix it with the formalin. One thing needs to be warned about using the formalin. Remember, this chemical is carcinogen substance, and for who not really familiar with this chemical, actually this can cause a cancer. So you better prepare your goggles, gloves, so for your own safety. All right. And what the condition is very good to collect this, floating microplastic. Actually, the clumsy condition is you need to consider. You can check from several websites. Why you have to avoid the rough sea conditions, because it's very difficult to maintain even yourself on the boat, and working on the rough sea condition is not a really good. It is not healthy, actually, I would say. And then another phenomenon that you should avoid is a plankton bloom, because of as I mentioned, like we have to towing like 10 to 20 minutes, but we have some experience last time. We have some sulfur blooming, and it's just like 5 minutes and all of the net is getting clogged because of, many of planktons is there. So should avoid to towing during this time. And before you preparing your equipment or something, you have to make sure that your net is clean before checking the fill blanks. And also, once you're taking the net into your laboratory, make sure that area is really clean to keep your net equipment. All right. I believe that not all laboratories have a net to collect the sample, but they have different kind of equipment or tools that you can use for collecting the surface microplastic. For example, the pump, the bucket, the water sampler, and the net I just already mentioned. And this slide just showing you the pros and cons of each equipment. For example, if you use a pump, it's very easy. You can use and you can collect the water from the specific depth that you need it. But the disadvantage is very high cost, and also they have the high suction force, and that could affect your small particle of the plastic, I would say. And for the bucket, it's easy, it's cheap. You can conduct even using the bucket, but you cannot take like a 1,000 liter a day using a bucket, because it's like time consuming and using your manpower to do it. So, but it depends on the situation. So I'm not recommend to which one. It depends on your situation in the lab. So, and the last one, the net, I already told you, they can collect a large volume of the water and can use for the data harmonizations as well, because there are many people working on that already. And but they have some limitations. So because of your designing the size of the net that you will be used, for example, we use 330 micron of the net size. So it's kind of limited your particle size as well. That I will give you some idea for what kind of equipment that you use, so you can apply, to suit your own condition. Next, okay, I will finish the microplastic from the surface, and then I will move to the beach sediment. The beach sediment, it actually is very good location to study a microplastic because, once you're walking in the beach, you can see, the physical factors or the biological factors can be, uh, how to say, can be affect the accumulations of microplastic in the beach sediments. And the interaction with the physical force, such as the waves and currents, combined with the human activity, biological interact, can lead to the widespread of present microplastic in the beach as well. And yeah, I think it's worth it to looking the microplastic on the beach, as you can see, what the factor can be involved with this microplastic accumulation in the beach. How to collect it? So the beach sample is quite simple, but you need more people to help you with that. I will explain you a little bit what where is the best for collecting the microplastic on the beach. As you can see from the slide, so the back shore or the back beach area. So actually, this area is just a beach extend from the limits of the high water for foam lines to the dunes or extreme inland limited of the beach. So in this area, we consider to collect the microplastic around the beach. The second figure can show you how to put the line transect. If you already determine, where is the rack line along the back shore, so you can place your the tape, around 100 meters along, the shore like this. This is just very good example how to put the quadrat. The quadrat, actually, I recommend to avoid using the PVC or something that made from plastic. I recommend to use the stainless or another kind of non-plastic material, so it will be less contaminations from this kind of equipment. All right. What you're gonna do to collect your your sediment sampling? First, you identify the area, place the tape, and put the quadrat like this from 0 to 100 meters. And then maybe some of you have some questions asking like, how, how the distance from each quadrat. So I would say you can use this randomly. They have some website, you can just click a random number and you can place your quadrat in each number, the meter number along the line transects. And then you're using the metal scoop like this, collecting the sand or the sediment, from the top and digging up a little bit, like the depth approximately 5 centimeter or 15 millimeters from the surface to, the bottom of the sediment, just only 5 centimeters. And then you're using a sieve. The size of the sieve that you're using for the beach sediment, it depends on your purpose. Some of them can use just only 5 millimeters and 1 millimeter. But, it depends on your purpose. If you need to looking for a smaller microplastic, you have to use smaller size of microplastic, smaller size of the sieve, sorry. And then you can use I take a metal screw from the bucket if you collect your sediment in the bucket. So take it into the sieve and using some filter water, to sieving it. And then some return sample will be examined in the laboratory later. For example, if you would like to see a smaller fractions or smaller microplastic, then need more steps to digest and, to extract your microplastic from this sediment. All right. I'm not clear each any part, please let us know. I will clarify with this later because my time is quite limited here. Yeah, how okay. So I have some times. And yeah, please avoid any plastic materials from your tube. For example, the bucket, please use different kind, stainless or whatever, not a plastic one. This is just an example. If you have more people, you can, extend your area. Actually, is good, but it's time consuming. But the advantage is you can compare and collect your data for further comparing to another work as well. Actually, I recommend if you have more people to help you with that. All right. So floating microplastic and microplastic in the sediment already been mentioned. Next, we're going for the seafloor sediment. When we're talking about the seafloor, it's not just only the deep sea that we have to consider. Actually, seafloor can be, in the coastal area, the shallow waters as well. If it is shallow, like less than 30 meters, so you can use a diver to collect the sample using a push core or whatever you have, or the grab. We also use it once your sediment is below water. So we can use this kind of equipment to collect it. And for the deep sea, actually the deep sea is very challenging unless you have the ROV or the vessel that is already, investigate with the deep sea environment. So, and that it actually is a challenge for the people working the deep sea right now because they need a standardized protocol after they analyzing, the biota or even the sediment from that environment. So yeah, this is kind of basic thing that just showing, and you can have some idea with this. All right, then I move to the marine biota. As I call myself a marine biologist actually. So I quite familiar with like marine behavior and stuff. This slide just sharing you before you going to conduct any research about marine biota. Please consider their behavior. So, for example, in this figure, you can see actually this is different kind of organisms from plankton, from nekton, and to the benthos. So this different kind of habitat can reflect something for the behavior. I mean, the feeding behavior as well. So, but believe it or not, all of them, I mean, their body are contaminated with the microplastic. All right, as I mentioned, especially the planktivorous fish, for example, most of the papers saying this kind of fish is contain a lot of microplastic because their feeding behavior. For the pelagic fish, because they are living in, like kind of surface water, most of them, they found the low density particle as mentioned because low density particle is floating on the top of the surface water. And for the benthic, of course, if the polymer is denser, you will be dropped down and accumulate in the sediment area. So, that can give you some idea what you're going to do with your biota. All right. Why we need to study the microplastic in the marine biota? Actually, it's obvious because at the end, who will get the effect of this? Human, right? And we need to study the microplastic in marine biota because it could harm to marine organism itself. And if they are contaminated, is especially with the commercial or eatable fish, for example. So eventually they will, accumulate in the human. So it is very risk to the human health. And right now, since like a couple of years ago, they have some published saying, they found the microplastic in the human blood. So it raised more concern about, how to investigate the microplastic in our environment, especially something that we eat it. Microplastic not just only eating and accumulating in our body by ingestion, but actually from, like when we breathe, it's in the air, it's in the water, it's everywhere. So it's really need to be concerned. And yeah, some of the species, for example, the polychaete or whatever, that can be the bioindicator, especially when that area can have, the high pollution or high hotspots of this kind of pollutants. So they can be a bioindicator of that areas as well. So at the end, for this kind of study, so I think it's a good idea that to show the people what we have, what we get from our research, to raise the topic, raise their concern, what they will do for reducing the plastic in the future. So, all right, so I almost done. Actually, this is kind of my last slide. So maybe some of you not familiar with the marine environment, marine biota, but I share some idea how to collect them. I'll start with the plankton. The plankton is actually some small stuff, even the phytoplankton, zooplankton, from the ocean. So we can use some several kind of equipment to collect it, the plankton as you can see at the beginning of the slide. And the skin bottle. You can also use this kind of equipment to collect the plankton. Next, the nekton. Nekton is something that they can swim by themselves. For example, this kind of fish, the fish trap, the fish net, whatever, or even you can catch them by your hand if you can. But yeah, I think it's a little bit difficult. All right. Then how about the benthic organism? So as I'm working on coral, so sometimes I diving and collect the sample by myself, but some of them, they can use another kind of net, throw net or whatever, to collect some of the marine benthic over there, or even the crab also you can use. For the deep sea, I cannot dive into that depth because it's too deep and the human cannot reach it. So basically they use a net as well. They have a deep net that can collect the deep sea creatures. And also the ROV, I'm not sure that you're familiar with this, but actually is an equipment that they can attach with some optional stuff and then can collect your samples as well. And also using a grab the coral, something like this. So it's the last one, and the last one is kind of marine mammal. So I read some of the publications about how they study microplastic in the marine mammal. Actually, it's very interesting. Basically, you cannot kill them, right? So basically, you have to collect something else from them, which is mean could be their feces or something else, or after they die, maybe you can, see what they ingested. But basically, uh, we use the handy net, together with the drone to survey around the area that this kind of animal living. And if they excrete some of the feces or scats, so we need to collect it immediately like a fresh one, because otherwise we'll be diluted with the seawater. Or if your marine mammal is living on the shore, you can even, following them and collect it by your hand as well. Keep it the safe container. So yeah, that is some idea to how to collect, monitor the sample from the floating microplastic, sediment, seafloor, and marine biota. I think, yeah, I finished my first part. Right. Any questions? I'm not sure.
Thank
I
you.
stopped sharing my screen.
All right. Thank you, Mei Sensei. So
Thank you.
from now, we started to Q&A session for the monitoring part. If you have any question, please type it in a chat box. And we already have 1 or 2 questions from the
All right.
participant. And first question is, volume calculations are a big source of error. So how do you guys handle uncertainty in determining this parameter? I guess this is for the net sampling.
Sorry, I cannot
Can you
see.
see? All right. And during the Newton net sampling or Plankton net sampling, the how to calculate the water volume is important to explain the abundance of plastics, right?
Yep.
So how handle this uncertainty matter? Maybe this is a question for Belén Sensei. She is a zooplankton researcher, so she has a lot of experience. Can you answer this question?
Yeah, sure, no problem.
Yep.
I am very familiar working with plankton nets and also Newton nets, but most of the time what we use, for addressing the volume calculation is the best practice is to use a flow meter. Every flow meter, the manufacturer can give you a formula to address the volume calculations. It might differ from one company to another. But basically, the best practice is to use in a flow meter. Then you can calculate the cubic meters. These devices, the flow meters, have some kind of error estimated, but also the suppliers, the company suppliers offer you some estimation of the errors of by the manufacturer, or you can also calibrate the flow meter by making some trials in your laboratory before going to survey. But I think this is the best approach. Even so, the flow meters can lead some error, but no device is perfect to measure exactly the volume filtered by the net. But one thing we should consider, as Mei Sensei explained before, is avoiding the net clogging, because once the net start to clog, means that the volume calculation and the filtration by the net is not accurate anymore. So you have to avoid that every time. I think that clarifies your question, but I will, if you have something more to ask, please let us know in the chat.
Yeah, sure. Thank you very much, Belén Sensei, about these clarifications. I think we probably have more questions, but I can't see it. Sorry.
And another question is from the when you register this webinar, we ask everyone to share your question to us. And from the process, we have one question like, how much sample volume do you think is considered enough to correct?
Yeah.
All right, I can call. In case of the plankton net sample, 20, it depends on the targeted size. 20 minutes towing with 2 knots. This is a kind of standard to correct the sample. So we can't say exact water volume because it depends on the water current speed or the situation. But 20 minutes and 2 knots. This is a kind of standard. And in case of glove sampling or in case of a small plastics, according to your analytical method, perhaps 20 liter or 100 liter is enough because we can't correct all of the particles on this very fine filter paper. And actually, we don't have kind of standard volume to measure the plastic. And we need to have standardization or harmonization about this. This is a fact of the microplastic research in the environment. And in case of a wastewater treatment, maybe the volume gets less because we can't handle all of the water.
Yeah. Thank you very much, Nakano and Belén Sensei, about to answering these questions. I think not just only the surface water or the sediment that we didn't have any kind of standards of how, I mean, in terms of this surface water, they have kind of exact number that you can really know, like a water volume that you can receive, that you can collect. But in terms of the organisms, I'm not sure that you are working on this kind of marine biotas as well. But for example, if I'm working on corals, so I cannot answer that to like how much is considered is enough for collecting the corals, because this animal is kind of strict, they have some different rules to collect it. So yeah, I think maybe if we have some platform to share this kind of idea, I think would be good too. I think, yeah, my time is up. Let's move to the next topic. Okay, our next speaker is already here, Dr. María Belén ALFONSO. As I mentioned, she is currently working with the ingestions of microplastic by zooplankton. But right now oh, I have to stop sharing my screen. Can you share your screen, Sensei? Yeah, and right now she will share with you about the sample analyzing and processing. So I think if you have any questions about this during she trying to explain, please let us know through the chat box as well. I think I will give this floor to Belén Sensei, please.
Thank you very much, May. Can you see my slide?
Yes. Yes, we can.
Thank you. Thank you very much. Okay. My name is María Belén ALFONSO. You can call me Belén. I'm from Argentina, but now I'm working in Kyushu University a while ago. So today I will introduce sample processing and analysis for microplastic samples. It will take around 30-40 minutes. And after that, we will have a small Q&A. And after that, you can have a break so you can relax a little bit. So let's start. Okay, once May Sensei already explained very good how to obtain different type of samples for microplastic analysis. But what is next? Once you have your sample, you have to wonder how you retrieve your microplastics, right? So you need to understand what kind of sample you're considering, right? The most important question is the material content in your sample. For example, we have different type of samples with a considerable amount of material content in the case of biota samples, sediment samples, sand samples, and others with not much material to digest, like water samples, or much less in the case of air samples, for example. But for each kind of sample, we need to address some questions before understanding how to proceed with the sample processing, right? For example, biota samples, we need to understand which is the size of your sample. Is an organism, is a colony, is a plankton sample with many individuals on it. It's hard structures on it or not. Do you want to digest and analyze the partial part of the organism, like the stomach or any other, or the liver or other parts, or you want to digest the full body? For example, in plankton samples, we digest the full individual, right? In the case of sediment samples, soil samples, or sand samples, you might have some characteristic features of the samples, like a lot of inorganic content or also a lot of organic sediments on it. In the case of water samples, you might find a lot of plankton content or not, depends on the sample. For example, open ocean samples might not have much plankton compared to coastal or estuarine samples. What about the inorganic particles on it? Do you have some or not? Maybe river sample has a lot of inorganic particles and maybe open ocean not much. And what about the organic particles? You can also find, instead of plankton, you might find some small jellies, small fishes. So you need to consider all this before proceeding, right? And in the case of air samples, maybe it's the easiest and the cleanest kind of sample, we should say, because you might find some inorganic particles or organic particles, or maybe an insect or something. But it's not big deal to sample processing, maybe compared to others. So considering this, we have to say that there is more than strict categories, is a spectrum of different situations. So for every situation, you need to consider, what should I do next? How should I consider the sample processing? So in this presentation, I cannot address all the types of samples and all the type of conditions, but I will try to summarize for biota, sediments, soils and sands, and water samples as an example. But yeah, remember that every condition is different and you might need to consult more about your particular sample later. So, sample types, we say that we can have water samples in seawater, freshwater, wastewater samples, soil and sediment, biota, air. Once we consider the sample condition, the following is to consider what kind of chemicals you will use, considering the conditions of your sample, as explained before. Also, which are the steps you will include in the digestion, which is the temperature, which is the duration, which are the replicates you will consider for the digestion process. After digestion process, we will start following by density separation. Is there any inorganic matter left in your sample? You will consider a density separation. Density separation is a process we can use to make the inorganic particles to get in the bottom of the sample, so the microplastic will float because of differences on the density. So you also need to consider what type of salts you will use, the density of the solution, the duration of the process, and if you're going to make repetitions. After density separation, you will wonder which is your particle size target, because the process will not be the same, the following process, if you're focusing on small microplastic instead of focusing on large plastic particles. So after you decide which is your particle size target, you will choose the equipment, the filter types, the vacuum pressure, the sample volume you will use for the manual sorting and the filtration process. And finally, we will finish with the classification and quantification, where you can also address what kind of equipment you need, and you will characterize your particles by size, color, shape, number, and polymer types. So first of all, let's summarize a little bit about the best practices in the lab before starting sample processing. You might find yourself in your laboratory, everything looks clean, everything looks neat and tight, but you will have microplastics in the air, in the surface, in any element, because plastic is everywhere in our daily life. Even in our clothes, you can find plastic fibers, or even in your materials. So even you're very clean, you need to check on this. So the best practices to avoid the contamination of your samples include avoiding plastic materials as much as possible, using lab coats, using a clean bench if possible, cleaning the surface, clean the material and the reagents, and reduce people's transit in the laboratory while you're doing your sample processing if possible. And safety first, always use the safety elements. But you can see more details about the QA and QC in the following section with Dr. Nakano. So this is an overview of the equipments we will use, you can use for the sample processing. This includes a fume hood, a clean bench. A clean bench is very highly recommended, but sometimes in your lab you will not have one. In that case, you can use a fume hood, but always to reduce, the particle exposure of your samples, right? But always use it turned off, because the air circulation of a fume hood is different from a clean bench. It comes from outside to the inside. That means you will, if you turn it on, you will bring a lot of particles and fibers to your sample. So, but it's better to use a clean bench if possible. Also, between the tools, try to avoid the plastics. And for the reagents, you will use several chemicals, including milk or distilled water to filter, to rinse and wash your samples in many steps. If you don't have a milk device, you can use distilled water as well. For this, or any reagent you are going to use during this process, you need to pre-filter everything. That means you need a filter equipment and filter glasses to remove any particle contamination that could be in the reagents from the manufacturing process. Even the chemical reagents are like chemical grade, very good grade. It might have some microplastic on it, mostly small microplastic that you will not able to see. And of course, you will need a lot of consumable goods, mostly important is the filter types. And the protective equipment, including the lab coat, the glasses, the mask, and a trade glove to avoid accidents. So let's start with the digestion. Once you have the sample, let's talk about, for example, a Newton sample. You have your sample with a lot of plankton, a lot of particles, maybe some microplastic on it, some sediments and organic content that you cannot say what it is. You can use different type of reagents to eliminate this organic matter from the sample to retrieve your microplastics. One of the most used reagents are hydrogen peroxide for wet peroxidation at 30%. It's very effective for vegetal tissue, and you can improve and make it quicker reaction using the Fenton technique. But I don't recommend the Fenton if you're very initial or beginner in the digestion, because it's very reactive and you can have some problems to manage, but it's easier using just the peroxide, but it takes a little bit more time. Another option is using KOH or NOH, potassium hydroxide, sodium hydroxide. This is very effective for animal tissue, and the digestion has good results as well, same as wet peroxidation. One of the most used in the papers nowadays, but usually at the 10%. In the case of enzymes, it's very good efficiency when combined, and it's very specific. For example, you have protease, amylase, lipase for lipids and others, but it's very time consuming. So maybe also it's not the easiest approach for one person being a beginner, or if you don't have too much time, or you have a very high number of samples. Using enzyme is most costly. I mean, the costs are higher and it's very time consuming. Also, you can combine, for example, combining wet peroxidation with enzymes, combining two-step digestions using wet peroxidation and sodium hydroxide. For these 2 or 3-step digestions are more effective than one-step digestion, mostly when the samples has a lot of different animal, vegetal tissues. So that is time consuming as well, of course, more than one step. And finally, you can use some acids like hydrochloric acid or nitric acid, but always these acids could be very aggressive. It's very effective to eliminate any organic content, and it would take less time, but you need to use it only when necessary, because the acids are more aggressive. From the questionnaire results, when you register to this webinar, I found out that, for example, from the first 200 registered participants, most of you deal with water samples, sediment samples, and biota. And most of you are applying already wet peroxidation or alkaline digestion, or 1/3 of you might have no experience, but that is not bad, because you will learn today a little bit more. When we apply in these reagents for the digestion steps, we need to consider one thing, that is the temperature. In the case of wet peroxidation, it's highly recommended from previous studies not using temperatures higher than 60 degrees. If you increase the temperature or the concentration, you can damage polymer types, for example, polyamides. In the case of KOH at 10%, it's recommended to use temperatures below 40 degrees because also can damage polyethylene, polycarbonate, and PET at higher temperatures. In the case of enzymes, these are very specific and no damage was observed to the date for the microplastic, so it's very safe to use enzymes, but as I say before, it's time consuming, so maybe you need to consider which is best. And in the case of acids, is the most, I mean, the acids can affect a lot the microplastic particles, so it's highly recommended to use at room temperature, not increasing the temperature to make it quicker. Because can damage polyamides and other plastic polymers, including high-density polyethylene, low-density polyethylenes, PET, and others, according to previous studies. But always, if you want to know 100% for your protocol, if will damage or not your target particle size for your study or your polymer type that you're interested in, it's better to make this kind of trials in your lab and see if the temperature and the conditions you're using for your sample digestion can damage or not the plastic polymers or the size or the structure of the particles, right? But this is the recommendations from the cited literature from previous studies. One other thing to consider is the duration. The duration of the digestion process is highly variable, as same as the condition of the sample. So we cannot say a specific time duration for any sample, but always try to apply a precautionary approach, less time as possible, until no remaining material is seen in the sample or no much material. If you already can filter or pick your particles and is enough, it's okay. Always follow the recommended temperature and concentrations for each chemical. And I recommend to use replicates, but this will increase, of course, the duration of your digestion process, but you can find a balance between time and data quality. At least 2 replicates is recommended. If you can do more replicates, it's better. For example, in marine samples, water samples with a considerable amount of plankton in coastal areas, for example, you can use a 2-step digestion, applying wet peroxidation with phenton 60 minutes, and after that, 10% KOH for 72 hours at 40 degrees. Sample preparation. So, for the digestion process, now I will explain more in detail the step-by-step if you have more questions about the details, but this is a kind of example for a water sample, right? As I said before, this will change for any particular sample, but we can discuss it if you have any questions about your particular sample. So if there is too much debris in your sample, you need to work in several steps or fractionate your sample to avoid overflow. Always open the sample under a laminar flow hood, turn on, or if you don't have a laminar flow, you can use a fume hood, but as I said before, please turn it off. Remove any leaves or wood residues that you can find that is not of interest for you, and using tweezers, and you can rinse them many times using milky water so the water drops in your sample if any particle is attached. And then you can filter the samples using a glass panel and a nylon mesh, or a stainless sieve with a mesh opening smaller than your target size. After that, using a washing bottle, you can rinse the inside of the sample container to avoid any particles to be attached and you lost those particles. So rinse many times with milky water or distilled water and pour the liquor in the funnel or the sieve along with the rest of the sample. Then you can pour the material retained in the sieve into a label beaker. Rinse with milky water until no material is in the sieve. And after that, please check carefully under the stereo microscope or with a magnifying glass the final condition of your mesh, because you might find some particles attached to your mesh or the sieve. After that, we will process with the digestion. In this case, we can use, for example, wet peroxidation. You need to filter the regions before use to avoid any contamination. You can add the wet peroxide to the beaker until the sample is submerged, and place the beaker in a heating plate or incubator or a water bath at less than 60 degrees. Then you cover the sample always with aluminum foil or a glass to avoid the contamination, and always check the temperature regularly to avoid any increase in temperature and damage of the particles, okay? And as the liquid starts to evaporate, please add more hydrogen peroxide as needed until organic impurities are eliminated or reduced to a negligible level. After you finish, you need to remove the sample from the heating source and cool to room temperature. When finished, there are two options. No residue is left. That means that you can filter your sample using a filtration unit and a chosen filter type under the laminar flow cabinet. You can use multiple filters as needed. This is to avoid the sample layers to form in one filter and the particles be trapped between layers. So please always try to keep the volume reduced. So, sorry, I received some message. If there is any inorganic residues in your sample, you can extract the peroxide solution or the reagent solution you choose for digestion by filtration using a nylon mesh or a sieve under a laminar flow cabinet. Then rinse the sample with a washing bottle with milk or filters distilled water and wash the sample back into the beaker using more water. And then you will proceed to the density separation step. This is only necessary if you find that you get inorganic particles remaining in your sample. But if any particle, the density separation is no need. Sorry. So for the density separation, we have two types of equipments, or there is more types, but this is the most commonly used. You can use a funnel for separation or a glass beaker. Glass beaker maybe is easier for you if you're in a beginner step, and using a funnel for separation with the funnel support, a clipper, and a rubber tube to close the end of the sample is better. In this device, you will pour the sample with the saturated solution. In the case of the density separation, we can use different types of salts and densities. As we know, the specific density of the plastic particles range between 0.8 and 1.4, but this depends on the polymer type and the manufacturer. So as we want to separate these particles from the heavier inorganic particles, we will use a density separation. For this, we can use salts, different type of salts. For example, we can use zinc chloride, sodium iodide, sodium dihydrogen phosphate, or sodium chloride, among others. This is the most common examples used from previous studies. In the case of zinc chloride, the density is quite high, it's 2.9 grams per cubic centimeter. So you will have a high recovery rate. As much higher is the density, you will be able to retrieve more particles from your sample, more polymer types. But in the case of this salt, it's very expensive and is toxic and corrosive. So you should be very careful if you decide to use this one. In the case of sodium iodide, the density is lower, but it's still good. It's 1.83 for a saturated solution. So you will have a good recovery rate, but also it's expensive. In the case of sodium dihydrogen phosphate, density is 1.69. So the good recovery rate as well is not toxic for the environment and is low cost. So it's highly recommended to use. And the cheapest one and most affordable and easy to obtain is the sodium chloride, but the density is 1.2. That means that even if it's low cost and environmental friendly, some particles or plastic polymer types will not be recovered in a 100%. Maybe you will get some of them, but not all. So you might load some particles like PET or PVC, which are heavy density plastic polymer types. When it comes to the duration of the density separation process and the repetitions, is highly variable according to your sample characteristics as well. But it's recommended to make the sample density separation process at least 3 times. And repetitions depends on the quantity of inorganic material. For example, sand or quartz samples, you will find a lot of inorganic material, and you might need to make it more than 3 times. So for the process overview, you start preparing a solution. For example, if we use NaI, we can pour 184 grams of the salt in 100 milliliters of distilled water or milky water to obtain the saturated solution. Once you prepare the saline solution, you need to filter using a glass fiber filter or similar to remove any potential contamination particles from the manufacturing process. After that, you will take 100 milliliters, for example, of saturated solution and put in a sample beaker, then mix with the sample, right? With the remaining sample from the digestion from the previous step, and mix for 10 minutes and let it settle for 1 hour. Then you will collect the 1/3 supernatant using a glass pipette. And in the case of large particles that you can already see by your eyes, you can also pick it by tweezers and preserve for later. But if you're collecting only the surface supernatant and you see no particles by your eyes, but maybe there, you keep it in a new beaker. Then the remaining liquid, you will repeat the steps like 2, 3, and 4. This means you can add the saturated solution, mix, and settle 1 hour. And then again repeat and recover the 1/3 part in the surface and mix it with the previous one you recover. That you will repeat it at least 3 times. Then you will have 1 beaker with the recovery 1/3 parts all together. And you will filter this obtained sample using a filter equipment and a filter inside a laminar flow cabinet. You need to use, same as the digestion, as many filters as necessary to prevent any thick layer forming. But after density separation, we are not expecting to have much more material than the plastic particles and maybe another type of particles remaining, but still no much content, right? And then you can place the filter in a labeled Petri dish and allow it to dry at room temperature, or you can use an oven, but using low temperatures like 40 degrees for a few hours and let it. So the final step is the manual sorting and filtration before the plastic characterization. Here we need to consider one thing. Most of you or many of you might considering only analyzing large microplastics. Large microplastic is a term to refer to those particles above 300 micrometers or 500 micrometers, which are very easy to see by your naked eye, but depends on your visible conditions, of course. But that means that you can do manual sorting with tweezers under a stereo microscope or using a magnifying glass with no problems. But if your target for your study is small microplastics, that means that you're focused on particles below 300 micrometers or 500 micrometers. It's hard to see by naked eye. So you need to filter the process samples, as we said before, after the digestion or the digestion and density separation. And you need to inspect your filter under a stereo microscope or using a microspectroscopy equipment like micro-Raman or micro-FTIR, which is more efficient. But if you're going to study small microplastic and using a microspectroscopy, it's very important to consider what kind of filter you will use for your process for obtaining your last sample, right? In this case, we have different types of filters in the market, but for example, PTF filters are very good, are easy to obtain. It has a pore size around 5 micrometers, which it helps and it works good for small microplastics. You can find in 47 millimeters diameter. is okay to use for transmission mode, for example, that Nakano-sensei will explain later in more detail. And you can also, if you want to reduce the area of your sample, you can also use some stainless support, which is custom made, and we can share the details about the device in the picture in the right later with you if you're interested. Also, you can use gold or silver coat filters or aluminum oxide membrane like Anodisc, which are highly expensive. Maybe it's hard to get in the market, not so easy, not so accessible. But you will get very good results for the transmission mode to use in micro FTIRs with no problems. And in the case of fiberglass filters, which are the most commonly used for many things in any chemical treatments or laboratory processings, you can find them in 25 or 47 millimeters diameters. It's accessible cost, but it's good for manual sorting, but not useful to use in, for example, in micro FTIR for transmission mode. So you need to consider what kind of filter you will use for your final sample before filtering. And then we can follow and finish with the classification and quantification of your sample. You will inspect the filter for potential plastic particles under a stereo microscope, following a pattern like the one in the picture, making sure that you will analyze each area of the filter, okay? So try to take a picture of each particle if you have a camera attached to your microscope, and then you will describe each particle according to the shape, the color, the size, and the polymer types. But we will explain more about this in the following sections. So now I will summarize. Maybe this was an example for a water sample, a common water sample with some plankton and organic content. But what happens when you have a biota sample? In the case of biota samples, you might be working with individuals like fish, and a specific organ like a stomach content of a bird, or you're working with communities like plankton communities or coral colonies. So in the case of biota samples, for the sample preparation you need to consider the number of replicates according to the individual size. For example, if you're working with the stomach content of birds, you might need to reduce the number of replicates. But in the case of plankton samples, you will have a lot of samples from your plankton net. So you can use as much replicates as you want. As much replicates you use is better because your data will be more sound and will have less error. For the digestion, in case of a considerable sample amount volume, you need to fractionate. Also remember to avoid the use of acids and high temperatures or concentrations. Use acids only for calcareous skeletons, for example, calcareous content. If you need to eliminate the calcareous content, you need to use acid. You cannot avoid it, but always use a precautionary approach using low temperatures and low concentrations. And if you still see some visible remaining body parts, for example, some exoskeleton from the organisms, you can remove them with tweezers instead of increasing the temperature and increasing the concentration or the duration of the process to avoid particle damage. Always try to avoid particle damage, mostly for small microplastics because they're highly sensitive. And also can happen that you have some oil excess problems in your samples from some fatty fish or something like that. In that case, I recommend to use two-step digestions or maybe enzymatic digestion using a lipase enzyme, or also a good technique is to rinse with ethanol your filters to remove some of the oily excess. In the case of density separation, you can use it only if necessary. Most of biota samples, sometimes you don't need to use density separation because there is any inorganic or calcareous content remaining after the digestion. So in that case, you can skip. You don't need to follow all the steps mandatory. And in the case of the filtration, please use multiple filters, avoiding the layer formation because this will lead to underestimation of your particles. And in the case you are focusing on small microplastic, always use minimum vacuum pressure, because also the vacuum pressure that you use in your filter equipment could make the fragile small particles to break down in smaller and smaller. So this can lead to overestimation, or you might lose forever your particle and you never see it in your sample. Also choose an appropriate filter according to the selected polymer detection method that you will use later, as I said before. In the case of sediment, soil, and samples, and sand samples, as Masons explained before, you can use a sediment core graph or sand soil quadrant to obtain your sample. And first, it's always recommended to determine the particle size distribution using a laser particle size analyzer. If you have one, like a master sizer, this is highly recommended. For the sample preparation, please consider to dry the samples at 50 degrees until they reach a constant weight before processing. After that, you can sieve the sample using, for example, a 2 mm to remove any impurities like wood or larger stones. And also, if some microplastic, you can see it there, you can also visually inspect and remove it before. Then, once you have your sample more clean, your sediment sample, you can fractionate your sample if you have too much volume, or use also replicates, which is highly recommended, and try to make at least 2 per point if possible. In the case of digestion, in case of a considerable amount or volume, please fractionate your sample to digest. Not put a lot of sediment in one beaker, because once you put the reagents and the chemicals will overflow and will be a mess. Also avoid acids, the high temperatures, or the concentrations, same as other samples. And for the density separation, you might need to make it more than 3 times, depending on the inorganic sample content. For example, you can put 50 grams of sample in 300 milliliters of saturated saline solution to work with. For the filtration process, please use multiple filters if necessary to avoid layer formation, minimum vacuum pressure as well for the small microplastics, and choose an appropriate filter according to the selected polymer detection time. Sometimes in the papers, you will read that people choose to make density separation before digestion, but I don't recommend this because some plastic particles might have some biofilm on it that make the density increase. So if you do density separation, you might lose some of these plastic particles in the bottom. And if you do after that the digestion, you might underestimate. So it's better to use digestion, remove any organic content, and then density separation. So a summary, please consider your sample conditions and particle size target before deciding your protocol. Use replicates to improve the data quality. And there is not exact protocol that works for all sample types, as I said before, because we will find many different conditions in nature. But you need to consider the best practices and always using a precautionary approach, which includes no high temperatures, which vary with the different reagents. As I said before, avoid high concentrations or strong acids when possible. The duration is highly variable, but always less as possible until, depending on your sample volume and the content of organic matter. Fractionate your sample if need to work in a more comfortable conditions, and use many filters to avoid underestimation of particles. So that's it. Now let's have maybe 5 or 8 minutes for some questions.
Thank you very much, Dr. Belén. So actually, it's kind of comprehensive summarize in terms of, the data, the sample processing and how to process your data. I mean, including the microplastic extraction. And yeah, actually, we have some questions, really interesting. They're asking about the high concentrations of the hydrogen peroxide, for example, like 30%. Once they use it, it could be decolorized, of the microplastic particle. So could you please explain how much we need to reduce the concentration of the peroxide for the sample?
Yeah, when you use wet peroxide, the concentration recommended is 30% solution.
Yeah.
So, what do you mean by reducing? I don't
May,
know.
may I answer instead?
Yep. Yeah, sure.
All right. Wait. Okay, good. I understand the meaning of the question. In case of
thank
the
you.
large plastics, if we use 8202, color of plastic get reduced, like a red color get white, and we can recognize if the particle has a completely artificial color, it is plastic, something like that. But if color gets white, maybe we can't say this is a plastic by color. This is a kind of the question from him. And last paper I read, it said 10% is recommended, but at the same time, color is not enough to identify whether the particle is plastic or not. So we need to use identification method instead of checking by eye. So if I answer how much we need to reduce the percentage, perhaps 10% is answer, but I recommend to use identification method instead of checking by your eye. This is the answer to the the question. Yeah.
Yeah, thank you for your comment. Now I get the point. But also, if you use or reduce the concentration to avoid decolorization, you have to deal with more increasing duration times for making the digestion work, right? To reduce the organic content. So sometimes it's recommended to reduce, but only if you have a lot of time to process your sample, because it might take a long time.
Exactly.
Yeah. All right. So
this is a clear example that there is no one solution for each type of sample. Yeah.
Yeah. For example, it depends on your samples as well. If they have a thick, like organic matter, so maybe you could not avoid using the 30% of hydrogen peroxide, but instead maybe you can use a little bit shorter time exposure. Maybe it could help, but not really sure what kind of your samples look like.
Yeah.
So you have to
also, as Dr. Nakano explained, the color is not enough to say this is a plastic. You always need to address the polymer type after that. So even if you lost the color, the color information of a particle for microplastic studies is important, but if I have to choose, I prefer the polymer types.
Yeah. All right. There is some other questions. Is it acceptable to use the calcium carbonate for density separation of fish muscle samples, specifically when the fish is fatty fish? You mentioned about some fatty fish before, I guess.
I mentioned that sometimes happen that after digestion, your sample might have some fatty content that you can eliminate using, for example, ethanol or using two-step digestions. I have to say I'm not aware of the use of calcium carbonate for density separation to avoid the fatty from the samples, but yeah, let me check after the webinar and we can maybe give you a better answer to that question. But now I don't have any specific information.
Yeah, because you have different kind of field, and then they have different kind of biotas or even other metrics, so it will be something particular. So, but we will keep these kind of questions, and then we will answer this later. All right. And I think it's for one last questions. Is it acceptable to use cellulose membrane filter paper for microplastic study?
Yeah, you can use cellulose if you're, but depends on as I said before, you have to choose the file type, the filter type. based on which process you will follow. If you want to analyze your sample by a screening method using a micro FTIR, micro Raman, you might consider what kind of filter is suitable for this. I think Dr. Nakano will explain more about this in details and you will understand better, but in the case of cellulose membrane for picking and sorting under the CO microscope is no big problem.
Okay.
You can use as well.
All right. It depends on your, ID equipment. All right. There is people coming. Which solution we can use during the density separation process? Wow, this is very interesting because, he asking about the atmospheric microplastic. So do we
Yeah.
have more time for this? Yeah, probably.
Yeah, I guess we didn't address particularly air samples, but air samples we know that might have not much organic content, but you can find some inorganic content on it. So I think any of the salt solutions I mentioned in the slides, you can use it for atmospheric microplastics process. I mean, as higher the density, you will retrieve more polymer types. But you also need to consider the cost and the access to the salts and if you can handle the toxicities and others.
Yeah. All right. Thank you very much for your answers. I mean, this webinar not just, like Q&A only. So if you have more comments or you have your particular own experience, please share it here. So it's kind of sharing knowledge, so instead of Q&A. So, and then we can have some, yeah.
Yeah, I think it's a very good suggestion because I think most of people have very particular situations, but maybe we don't have time to address all of it. But we were wonderful if any of you leave your questions and maybe we can answer after at the finish of the webinar or maybe another days as well.
All right, good. So do we have a break?
Yes, now let's have a break. I think it's 10 minutes break. And let's get back by 2:35.
All right,
No.
don't leave the chat,
No.
but
more because we extended.
Yeah. So let's see again after 10 minutes.
Yeah.
Okay, welcome back, everyone. So this is a third part of this webinar today. So Dr. Nakano Haruka will explain, a little bit more about of the QA, QC, how to deal with the microplastic in the laboratory. And then after that, we're going to have 5 minutes for Q&A as well. If you have any questions, please leave in the chat. After that, she will continuously about another session, which is the last session for the microplastic identification. So stay tuned. Don't go anywhere. And I think I give this floor to Dr. Nakano, please.
All right, thank you for introduction. My name is Nakano from Kyushu University. And in this section, for 30 minutes, I will explain about QA, QC, and LOD and LOQ and data description. Before we start, I want to explain one of the message from a kind of journal, a very brilliant journal, Stoughton, Science of Total Environment. And the editorial of the journal said about the microplastic research. To publish the paper in this journal, a detailed protocol of sampling and sample preparation should be provided. And also, the author should include the positive and negative controls. And the author should mention about the limit of detection and limit of quantification. And second, the Stoughton said identification of the polymer type is mandatory. So if you don't use any identification method, you can't, the journal cannot accept your paper. Also, to submit a paper, you need to compare your data international level. Otherwise, and you need to have a great discussion in the paper. This is an important message from one journal. And the Stoughton is one of the top 10 journal, top 10% of journal. But in my experience, another journals follow this message, and now to publish the paper is more difficult than before. So we need to consider these 3 points during our research. And this is a summary of the message. One needs include QA, QC information. And second one is mention the limit of detection and limit of the quantification. And third one is explain your method detail. And last one is using spectroscopy verification, identification. So in this section, I explain about number 1 and number 2 of this comment. So before I start about the QA and QC, we need to check the source of the contamination. It depends on your sampling processing or laboratory conditions. I want to give some example of the sources of the contamination. First one is paint particle from the bacell. If you use the baccells, first you can get the particle from the baccells, not from the ocean. And also during the sample processing, cross-contamination would happen. Also, the chemical you use perhaps has some particles. And during the laboratory experiments, the airborne contamination can be happened. And if your glassware has some dirty ones, first it produces a contamination, and your clothes can can produce airborne contamination as well. Also, makeup powder can contaminate your sample. These are the example of the contamination, and other than these contamination sources, first you have own contamination source, so you should check the contamination source before you start the experiment or sampling. And this is a summary of quality control. First, you should use a clean bench or do laboratory experiment here under the clean room, in the clean room, if possible. And also regular cleaning works to avoid the contamination. And using less contamination gloves and wearing a cotton lab coat also works to avoid the contamination. And here, the right side, you can see what should not to do. The first one is wearing face mask. First, face masks were made by the synthetic polymers, so it produce some fibers in the air. And also, I already said, to using the cosmetics should be avoided. Also, this is a suggestion to reduce the contamination from the glassware. I recommend to use ultrasonicator to washing the glassware. This is a step of the washing the glassware. For example, use for the first using the mild detergent, and after then using the sonicator, and rinse the equipment by the molecule or distilled water, and dry the glassware in a clean space. Perhaps using muffle effect works to remove the organic material. And after all of the procedure, you can cover them glassware with a clean aluminum foil. This is a basic idea to avoid the contamination attached to the glassware. And this depends on the laboratory, so please consider how to reduce the contamination in your situation. And so far, I introduced about the contamination, but Horton et al. 2021 said background contamination in the sample is avoidable. So it means we cannot avoid the contamination. So we need to check the blank negative control throughout the whole process of microplastic research. According to them, the key points of background contamination checkings are: number one, depends on the polymer type. It means the background contamination has dependency on the polymer type. For example, in case of their experiment, polyethylene, polypropylene, and PET particles were found in their background. But in contrast, PMMA or polyurethane, PVC, PA, PS, they don't find it. So we need to consider the dependency of the polymer type. And second point is, and contamination level affects more clean samples than higher microplastic samples. Please consider the situation when you treat the drinking water. If you find a 3 particle in the background data, but do you think it is okay? But also please consider that you find the 3 particles in sludge samples. I mean, the background for the sludge sample. Do you think it is comparable, or we can handle equal each other? We should consider the contamination levels in each samples and sample situation. And this is a basic idea how to do the negative control during the laboratory experiment. For example, place clean beaker and pour the non-particle water to there, and do lab work, same as the samples, and check the particle under the microscope and FTIR. The way to do blank test is not so difficult, but we need to do it. But so far, I introduced about the background contamination, but we need to consider the level of background contamination. And in this point, Horton et al. Introduced a limit of quantification and limit of detection in the abundance. These figures are the schematic of the LOD and LOQ. Left side, you can see 3 bars, and blank is in green colored one, and the number of particles we found in sample A and sample B is completely larger than blank. In this case, you can say sample A has some particle. But in case of the right side figure, the blank and sample A and sample B has similar values. In this case, you cannot say a sample A has this number of particle, because perhaps this is a contamination. So in this case, you need to check laboratory condition and should consider how to reduce the contamination and how to consider the level and difference. Some of papers suggest the statistical values, how to consider the blanks and the sample acceptable values. And in case of the limit of detection, 3.3 times standard deviation blank samples, if you find the particle larger than these values, you can accept the particle abundance. And in case of LOQ, 10 times of standard deviation blank samples, you can accept the particle abundance from the sample. So according to this suggestion, so we need to do blank tests for at least 3 times for one research. And so far, I introduced about the QA and QC. And LOD and LOQ in abundance. But at the same time, we have another limit of detection in size. It means size distribution depends on the mesh size or your laboratory equipment. So if, in case of the left side figures, if the fragment or your particle, your sample is the same size of the net, mesh size of the net, first the particle can pass the net. So you cannot correct all of the particle. And last time I used 330 micro mesh net. We cannot correct all of the particle less than 1 millimeter. So we need to consider size distribution and its size limitation. And this is a suggestion, one of the suggestions we can consider about the limit of detection in size. It is mesh selectivity. You can see is a detail from the Tokai et al. 2021. The idea of this is the shape of microplastics affects the retention rate. And please see the light figures. In the upper panel, you can see how much particle we can correct by the mesh size of 0.35 millimeter net. And also in the below panel, you can see the result of the 1 millimeter mesh size net. And the solid line here, it means if we consider the particle length or the longest length of the particle, and dashed lines here, this one and this one, this is a data from when we can consider the shortest length of the particle. Then, in case of the 0.35 millimeter mesh size net, if we consider longest length, the particle having the small longest length cannot correct perfectly, and a little by little retention rate get increased, and almost a 1 millimeter particle, we can correct almost all of the particle. In case of the short length, the most of particle can correct when shortest length is 0.35 millimeter. So it means we report microplastic size in the maximum length or longest length, but retention rate can be affected by shortest length. So we need to consider how much amount of plastics by your situation with this idea. And also, limit of detectable size is depending on what kind of instrument you use. For example, if you use conventional FTIR, perhaps you cannot measure very small particle. So you need to consider the limitation by the instrument. But about this, I will explain it next section. And so far, I introduced about the background contamination and LOD or LOQ. But from here, I explain the recovery test. And I recommend to check the rate of correction by your laboratory conditions. And otherwise, perhaps someone's data, they correct the particle only 30% and report perhaps. And in this case, we can't compare the abundance in each research. So we need better to report how much particle you can recover from your laboratory experiment. So the method is almost the same as the blank test. But the difference is you need to add some particles to check the recovery rate. And after correcting the sample, you should check the number of particles you can correct and check the rate, recovery rate. And here, Horton also mentioned the recovery rate has a size dependency. This is the data from the Wang et al. 2020, and they distribute several sizes of the particle to the recovery sample, and they found the recovery rate have size dependency. So in case of a large particle, recovery rate is quite high, but in case of a small particle, the recovery rate gets increased. So if the situation allows you to do it, I recommend to check the dependence of the recovery rate with the particle size. And this is a summary of the QA, QC, and limit of detection and limit of quantification. And so far, I would like to introduce data description. I guess already the participant here has some experience and know how to summarize the data. So maybe this is kind of redundant for you, perhaps, but I try. Please forgive me to explain about this. And microplastic data has some basic information, including the shape, size, color, and polymer type. And in case of the large plastics, so if you focus on the visible particle, you need to mention about these 4 items. This is a maximum requirement. And in case of the shape, perhaps you already read some papers and you found a lot of variation about the categories. But here, we propose to follow the GESAMP 2019 definitions. The definition is using the mainly 5 categories. One is resin pellet, and second one is fragment, and form, and film, and line. And this is very easy to categorize in case of a sample shape. If it is solid, yes. Having edge, yes, it is a fragment, something like that. So you can follow this diagram and you can categorize your sample into 5 types of the shape. And also, Lucier et al. 2020, they proposed to harmonize the shape types and compare the data. But first, you can see the subcategories. And so this meaning of the subcategories is, in case of the fragment in main categories, it has subcategories of fragment and form. In case of fiber, fiber has a filament and phosphorus fiber. And in case of beads, it includes granule and sphere. And here, beads, I recommend to use a pellet instead. And Lucia recommend to use subcategories, but I recommend to use the category proposed by Gesamp et al., Gesamp 2019, because in case of the form, expandable plastic, it has different way to distribute on the surface of on the sea surface compared to other shape of type. So we need to different data to predict the motion of plastics. And also, performance fishing gear would be better to the categorize in line instead of fiber, because the fiber is from the synthetic clothes. So I recommend to follow the Gesamp 2019 categories. And this is a definition of the size. I don't read everything, but I recommend to record the longest length at least. This is a minimum requirement. And this, you can measure the size under the microscope if the particle is larger than enough to measure. And also, you can record the color of plastic. And I recommend to use 12 color classification. And for example, non-pigmented case, clear and opaque, and pigmented, white, black, brown, blah, 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 blah. And perhaps this information can work to identify the source of marine litter. And this is the end of the section. And these are the references I used for this presentation. All right, thank you so much.
Thank you very much for your great presentation, Dr. Nakano. So maybe now let's move for some Q&A. We have some questions from the audience. For example, Priyanka is asking how to create homogeneous suspension of microplastic in water for accuracy of the study. Should I, how to create a homogeneous suspension of microplastics in water for accuracy of the study? I guess it's for the recovery rate.
Ah, I see. It depends on the particle density. We normally use ethanol to make uniform situation of the particle and distribute it to the milky water.
Yes, right. The ethanol, because of the density, it helps to, compared to water, to put the particles in a very homogeneous way after ultrasonication, right?
Yes, I think so. But at the same time, if you focus on only the floating microplastic, you don't have to have a completely suspended, right? I mean, if your target is only polyethylene and polypropylene, not necessary to sink. The particle no need to sink.
I see. Another question is, what does it mean if the sample concentration is greater than the LOD, but less than the LOQ, limit of quantification?
Yeah, this is a resolution and how to say, 分解ノート. I'm not sure. I'm sorry, I can't say it in English. I
No,
mean,
it's okay.
Resolution and quantification. In my experience, I'm sorry, I'm prefer this is not a correct answer. I mean, direct answer of this question. I think limit of detection is enough to report because everyone can calculate limit of quantification. And detection, limit of detection. I mean, is if sample correctly greater than less than yeah, this is a point. This is a confusing point for everyone, even me.
Yeah, we can address this question later also by a private message or to Danielle maybe later. It's okay.
Yeah, but in my experience and previous discussion with other colleagues, limit of detection is enough for this time. Not necessary to explain about limit of quantification because we already consider the statistical, difference between the sample and the background.
Okay. Yeah, I guess as soon as you clarify this information on your results for the results analysis and discussion, I think is best, right?
Yep.
So another question that we also received from the questionnaire for the registration questionnaire is, what is your quality controls on a field survey? I think this is from the beginning. Yeah.
All right. So if the situation allow you to bring the water, pure water to the sampling field, I recommend to bring it and in a clean glass bottle or clean glass, clean container, and open the cover lid and put it during the sampling. This is one of the way to check the contamination during the field sampling. And also you can see the field sampling by washing the clean net in the sampling field. This is another way. But at the same time, if you focus on BO2 and you cannot check the contamination in the sea, right? So maybe in that case, putting the water near your place is one of the solutions to check the field sampling, field blank. Is it okay?
Yep, sounds good. I agree. And another question is, what is it acceptable for a recovery rate in general?
Recovery rate. This is a very, how to say this?
I guess, yeah, there is not a specific number, right? But if it's good enough, I think will be higher than 80, maybe.
Yeah, if we can correct the 80% of particle, it is the better.
I mean, there is no right answer for this, I guess.
Yes.
The fact is to inform others that is reading your publication or your results or your report that using your protocol, you get this recovery rate. So your results have some limitation in this sense. This is very important, right? To be clear about the limitations of every protocol. So there is no specific 100% perfect protocol. But as soon as you inform everyone about the limitations of the protocol you choose, is the best for data harmonization, and people can choose if their data is comparable to your data, for example.
Right, exactly. So reporting is the most important thing.
Right. Be very specific and clear about the report of information. And give as much information as possible. Right. So we have time for more questions, or
Last time, do you count everything? Maybe. All right, let's move to the next section. And we can, if you have participant have further questions, please type it in a chat box and we can discuss it later.
Yeah, sure. Let's do that. Thank you very much.
All right. So give me time. All right. And so from now, in this section, I explain how to identify the plastic type by spectroscopy, especially FTIR, because we don't have much time to explain every single method. This is a basic sequence from the digestion to the identification. And you should consider some points after the digestion, I mean sample preparation. Which instrument will you use for identification? And what kind of function will you be used for your identification, and also the size limitation of identification technique. And these 3 points are very important to decide what kind of filter paper you use to correct the particle and how to prepare the sample for identification. So you should consider 3 points before you start the experiments. And here I explain about the FTIR. FTIR is Fourier transform infrared spectrometer, and minimum detectable size, yes. In case of the micro FTIR, 50 micron to 20 micron is a minimum detectable size in practical idea. And some manufacturers said you can measure the 5 micron, but in my experience, 15 to 20 micron is a safety level to say that this micro FTIR can measure the particle. And also, in case of the conventional ATR FTIR, the larger than 100 micrometer is the detectable size. Of course, you can measure the smaller amount of plastic than the 100 micrometer by the conventional ATR FTIR, but it depends on your scale. So, practically, these are the limitations of the size you can measure by the FTIR. And also, micro FTIR has a lot of types, and a lot of manufacturers sell the FTIRs. First you can see this type of micro FTIR and FTIR. This is, it has to, in case of the left panel, you can see the main body of the FTIR, and right side you can see microscope parts. Also, some of FTIR has just only the ATR. Later I will introduce about the ATR, but ATR unit only. And also other manufacturers show micro FTIR having the micro ATR unit. So you should clarify what kind of FTIR you use in your research. Otherwise, the reader cannot understand the limitation of your method. This is the basic idea of the FTIR inside. You can see light figure, and here the light source. And in the FTIR, the infrared can radiate to the beam splitter, and here the infrared can divide it into 2. One is go to the fixed mirror and return to here, and the other one go to the moving mirror, and after the reflection, the wavelength can change. And return to here. And the infrared here and here can combine here, and one infrared can radiate to the sample. And moving mirror can produce several wavelengths of the infrared. That's why you can see several reaction to the several wavelengths of the infrared. This is a basic idea of the FTIR. And after the infrared radiate to the sample, molecular vibration happens. And it means the molecular component in the sample can get the energy from the infrared. And after passing the sample, infrared reduces its energy. So you can compare the strength of infrared before radiation and after radiation. And this difference is, you can see as an infrared absorption spectrum. And when the infrared radiates to the sample, you can see several molecular vibration. These are just example. So, for example, you can see stretching vibration with a symmetry motion. So this one is molecular bond, and one molecular go other side. This is other side and vibrate. And another one is here, asymmetry stretching vibration. And also you can see bending vibration, other locking or scissoring vibration. And a certain infrared excites a certain vibration. That's why you can see molecular structure by the infrared IR spectrum. And IR spectrum can say this is a potential of this material. And this is the example how to interrupt the spectra. You can recognize several vibration or several peaks from the polyethylene. Here, you can see a symmetric stretching vibration, and here around 28 something, you can see symmetric stretching vibration, and around 14,000, you can see bending vibration, and 700, you can recognize the locking vibration. That's why you can understand this is polyethylene. And to interrupt the spectral peaks to the material, you can perhaps use the library checking, like for example, spectral search or some companies selling the spectral libraries to check your data, to compare your data to the library data. But at the same time, you need to better to know how to interrupt the spectra by yourself. Perhaps the spectral search can produce several candidates. And to know how to interrupt a table of infrared absorption frequencies, this is important. So you can see, you can search this one from the website, and you can see more specific peaks and specific molecular bond. And this is an example of already I told you, in case of the polyethylene, how to interrupt the peaks to the polymer type. And here you can see checking the large peaks, CH, 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 CH. That's why you can say this is a potential of the polyethylene. But at the same time, you can see the negative values around here. And this, why it happens, you should know the basic idea why such situation happens. I mean, if absorbance is negative, it means the plastic radiates infrared. Do you think so? Of course, no. There are a lot of factors affect the spectra. For example, changing radiation strength during the warming up the light. For example, you measure the background. I mean, atmospheric condition, room condition of the to check the atmospheric room conditions, you need to check the background first. But after background measuring, and some time passed, and get another sample spectra, perhaps the light source can change the strength of the infrared. And in that case, the difference background and sample has some, how to say, some kind of the trouble. So in that case, you need to measure the background again. Also, and perhaps humidity affect the sample situation, measuring conditions. And also, if you rest to the chamber of the FTIR, it affects. So you need to consider the situation, measuring situation as well. And perhaps some sample remains on the stage, and you measure the background there as you measure the spectra as a background, and the background has a signal of other sample. It means you can see the different, weird spectra. So you need to check whether the background and your spectra is okay or not. And also, some interference are there. For example, some of the method of FTIR cannot measure the carbon black, and also presence of water affect to the sample, sample spectra, and you can measure the mineral instead of plastic, and sometimes you measure the protein, and the protein is very similar spectra with the nylon, so very difficult to identify, decotonize which one. And also fatty acids is a interference of the microplastic, and performance degradation of the plastic can affect the quality of the comparison. All right, so let's try to interrupt the data together. And you can see step one and step two here. This is a polypropylene spectra. And in case of this, you can see stretching vibration around here and bending vibration here. But at the same time, you can see large peak here, 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 here. So in this case, you need to check small particle as well. And if you compare the data and infrared spectral frequency table, and you can find here is the signal of CO, and here is COH, and here is OH. So you can say this is a kind of degradation, or BO2 signals are there, something like that. And so far, I introduced the basic idea what is our FTIR and how to interrupt the data. And from now, I want to explain the functions, the method of the FTIR. Mainly, you can see the three streams to measure the sample. One is ATR. This is attenuated total reflectance method. And the other two methods are diffraction mode and transmission mode. So you need to know which method you used for identification, because it is important to say the limitation of the detectable size or data quality. And this is a basic of the ATR method. The picture shows a conventional ATR FTIR. You can see the silver stage, and in center of this, you cannot see, but there is a diamond prism, and you can put a sample to there, and you can attach the sample to the prism. This is a schematic of the sample processing. And in this method, the light source can radiate from the bottom side or perhaps upside, and the reflection happens between the diamond prism and the sample, and during the reflection, the infrared can excite the molecular vibration here and reduce the energy. And mainly this method can be used for visible particle. And next one is transmission mode. Mainly this was used for micro FTIR. This method analyzes molecular components by the infrared passing the samples. The light figure is a schematic of this method. You can correct the sample on the filter paper, and the infrared can radiate from the upside or upside toward the downside, and you can measure the difference from the light source side and detector side. And I'm sorry, it has, according to this method, the filter paper can affect the infrared spectra as well, because if filter paper can absorb the infrared as well. Also, size of the particle can affect. For example, the sample thickness is very large, and in that case, all of the infrared can absorbed by the sample. So spectra can be saturated. So you cannot detect the spectra, specific peak. So the sample thickness should be enough to thin. And these are the schematic figure of the spectra when the sample is too thick and the acceptable case. And the last method is the reflection mode. This method analyzes the molecular components by the infrared reflected on the sample surface. This is a schematic of the reflection mode. And from the light source, you can radiate the infrared, and on the surface, infrared can reflect and return to the detector. In this case, no interference from the sample, so you can see only the signal from the sample only. But you need to consider how amount of the infrared can return to the detector. In case of the left figure, the rough surface particle, even we try to radiate the infrared, some of infrared can go elsewhere and cannot return to the detector. So it means the infrared signal gets decreased, and it leaves the low correlation coefficients when you compare the sample and your library data. So if we use this method, it is recommended to use for smooth surface particle. This is a summary of the FTIR and its mode and pros and cons. So it depends on your sample. It depends on your situation, but you need to know the limitation of your function you use for identification. Otherwise, you cannot say this is enough to publish the data. And also, this is a kind of a small note. I recommend to check these items as well as the functions. Percentage you measured. 10% measuring, 100 measuring of particle, maybe the data quality is changed. And also library you use to compare. If you use one library, and if you use 3 or 5 libraries, maybe the results get changed. So you better to mention what kind of library you used. And I already mentioned sometimes, and which mode you use. For example, micro ATR, just ATR, reflection mode, transmission mode. It works to your paper readers to know the limitation of your technique. And also minimum detectable size. You can check by yourself and better to inform it. For example, aperture size or instrumental noise, something like that. And these are the basic of the FTIR measurement and identification. And Professor Raman spectroscopy has another notification. So you should check, depends on your technology you used. Okay, that's all. Thank you so much.
Okay, thank you very much, Dr. Nakano, for very informative data about the microplastic identifications. And I believe that we're not done yet. We have our QR code that, if you have your phone or whatever, you can access. Could you share the QR code, please? So yeah, we would like to get your feedback. So please, scan this QR code and, fill out your personal information. Especially, I think some of you may need the e-certificate. So please, I will give you some time to do it, or even you can take a photo and send us later and can fill out later. I mean, please make sure that you type it correctly. Otherwise, because this kind of automate response. So could you please paste the link here? Okay, I think that no sense.
All right.
They will.
Wait, give me time.
Yeah. All right.
So looks like in the last session, no question.
So I think most of the question is the previous sessions about
Right.
QA and QC, and most of them, I think Dr. Belén already clarified and addressed those questions.
Wait, sorry, temporarily I stopped sharing and I typed
I
it.
think there is a small question, but maybe you didn't address this topic, but maybe someone will find it interesting. Someone asked in the registration how to calculate carbonyl index. Not sure if you want to address that a little bit.
All right, carbonyl index. It depends several streams, and I cannot change the screen because now it's a feedback time.
No, yes, you can change the screen and then we come back to the QR code.
Oh, all right. The basic is the carbonyl index is a indicator to check the level of oxidation and oxidation. I mean, what is the degradation of the plastic is after getting the UV radiator UV light, the plastic release the CO2 and get the oxygen from elsewhere, from the surroundings. And a molecular component can change. Like in case of polyethylene, get the oxygen and having the molecular bond like a C and O. And the carbonyl index is the ratio among the amount of the CH molecular bond and CO molecular bond. And you can see the absorbance difference, absorbance level of each molecular bond, and you can compare the data, get the ratio. This is a ratio. I'm not sure it is okay to explain as an answer.
I guess, yeah, just a short reference about carbonyl index is enough at this point. Also, we have a lot of questions about FTIR during the registration, but I think most of them were addressed with your presentation. Maybe someone asked, which technique should I use for each range of plastic particles? Less than millimeters, which technique? And less than 10 micrometers, which technique?
Less than
Maybe.
4 millimeter, if it is visible particle, it's easier to use conventional ATR FTIR because we can pick up the particle by the tweezers and put it on the stage and just press the sample to the stage. So this is a very basic and easiest way. And in case of the 100 micrometer particle, I recommend
No, 10 micrometers.
Yeah, 10 micrometer.
Small microplastics.
Very small. Micro ramen is best idea to use. This time I didn't explain, but for
no, but yes, I agree. I think for less than 10 micrometers, I think the resolution for micro raman is best than micro FTIR. But you're looking particles up to 20 micrometers, maybe micro FTIR is okay, right?
Yeah, I think so.
Yeah. From my personal experience using the micro FTIR, actually the largest, the smallest size that I can detect is just approximately 25 micrometer. So I think that kind of limitations of the micro FTIR. But yeah, if smaller than that, maybe yeah, as you mentioned.
Yeah.
Yeah, I think most of the questions is relating to the QA and QC. I think yeah, it's very good attention because if you're considering about the publications or something, so this is the way that you have to plan before. So any more questions?
All right.
Sure.
We receive one question. Oh, okay. How do you place tiny microplastic less than 50 micrometer on FTIR to identify or the whole peak filter paper? The thing. In my experience, I recommend to use a filter paper. In case of the micro FTIR, and when you use a PTFE filter paper, PTFE filter, and we PTFE filter paper and Anadisk are quite expensive, but it has a small window to interrupt the absorbance IR spectra, but it can be used for other ends, so better to use such filters in case of less than 50 micrometer. But practically, I guess some researchers use tweezers or needle to put the particle to the stage and measure it. Similar method.
That's what I did actually. So once you identify what is the suspect microplastic, so using a file tweezer, and slightly move your particle to another same filter, and then using that to measure
Yeah,
using the micro FTIR.
yeah, but
Yeah, then it has some limitations because you can lose the particles. So it depends on how you prepare your sample. If you can make a good digestion and clean your sample properly to obtain all the particles in the
Yeah.
filter and then analyze directly from the filter, I think is best. But if you
Yes.
cannot do that because sample conditions is very particular or some reason, because could happen, yeah, you can also pick and measure one by one using the micro FTIR as well. But
Yeah.
yeah, you will need some high skills with the tweezers, some very good
yeah,
eyes.
right.
And
That's
yeah,
point.
try to avoid the wind, any wind currents in the room so
In the
you don't
room.
lose your particles.
Right. We got a message like I am requesting Nakano Sensei for another webinar on the carbonyl index. Is it possible in the future? Yeah, I think maybe
If everyone requested.
Yeah,
And
let's
Yes,
see
or we
about
can
that. I think it's a
share,
possible.
on request also. You can send us an email if you have any particular questions or any particular topic you would like to receive more information about.
yeah,
If we are able, we can send you some materials for you.
yeah. And
Exactly.
yeah, I mean, the important thing is, from my side, I mean, we share some knowledge we already know to the participants, but this is a kind of example. I mean, each laboratory, each university, each country has their own situation, and sometimes you cannot reach the facility, you cannot prepare all of the best situation, but as much as possible, you should adjust, right? You should find somehow to prepare, to have good data, and not only for paper, for your country. Otherwise, we cannot compare all of the countries' data. So, of course, if you use a micro FTIR or more advanced technique, it works well. But first, you don't have such techniques. You should do more basic and old-fashioned way first.
Yeah, I guess it's okay as soon as you can clarify the limitations of any approach you decide to use because it's the one you are able to do. It's okay, it's acceptable, of course, and it's good.
But
yeah, it's better to clarify.
And for better paper and for high rank journal, high level journal, they require us to be more international and more high level. To provide a high level data, something like that. So
yeah, I guess as much as you follow the best practices and you consider all the recommendations and you are very clear about your data limitations based on your protocol, I think the data is good. Yeah.
All right. I think it's time for closing these activities. This is okay. So if you have additional questions or something, you can access through this QR code, and then we asking you the last question, I guess, like additional comments or something. If you have more questions, please type in that as well. Oh, all right. So I think it's time to close this webinar. So let me say thank you for all of you today that's joining our webinar, Best Practice for Microplastic Research. It's been enlightening and productive session. I hope you found it informative and somehow, you can apply this kind of information to your current or even future research. I also would like to extend my deepest gratitude to our speakers, actually me included, for sharing the knowledge, the insight and experiences to all of us here. And last but not least, a big thank to our organizing team, especially to JICA-SATREPS for their hard work and dedicate in this, making this event possible. And once again, thank you, all of you, for your time and your engagement. Stay safe, stay inspired. And we're looking forward to seeing you at our next event. Not sure, it could be a carbonyl index or another topic. I think it depends on your request through the QR code. Thank you so much, really, and hope to see you again soon. Goodbye.
Thank
you
you,
so
everyone.
much.
Bye-bye.
Bye-bye.